Thank you very much for the invitation as a keynote speaker for 5th International Conference on Applied Zoology, ICAZ 2022. I'm very proud to be here to sharing my research with you. I'm going to deliver a lecture entitled Identification of Antimalarial Compound Isolated from Malaysia Soil, Streptomyces sp H11809 and their mode of action. This work is mainly done by my PhD student, Mr. Fauzi Mahmud. We are from Institute for Research in Molecular Medicine, University of Malaysia. And Fauzi also a tutor in Faculty of Science and Natural Resources, University of Malaysia Sabah, Kota Kinabalu, Malaysia. So this research also supported by Professor Lee Pinchins, also from Biotech Research Institute, University of Malaysia Sabah, Kota Kinabalu, Malaysia. So we're going to start with a research background. Malaria, as you everyone knows, I think is considered top 3 infectious disease tropics, cause than 250 million cases annually, and it's also cause 500,000 deaths annually. So it's considered a very deadly diseases. So second part is this is considered a this is, as everyone knows, is a mosquito-borne disease, and inflicted by 6 types of Plasmodium species strain, but Plasmodium falciparum, 95% of the cases, mainly in Africa, is considered highly mortality among children less than 5 years old and pregnant women. So this is a detailed cycle of Plasmodium, a life cycle, and its druggable stage. As you can see from different type of stages, it's come from the and this is show the old stage, the type of drugs that have been used. It can come from the old stages or blood stages or liver or blood stages. So if for further detail, I think you can read more in the others resources. So problem statement, I think, is for why we want to find drugs, new type of drugs. This emergence of drug resistance is Plasmodium, as one know, and second is low drugs diversity, and third is the antimalarial with unknown mode of action. It means some of the antimalarial drugs that really cannot distinguish what the molecule targets. So from the first one, emergence of drug resistance, as this is, as you know, they have since development of antimalarial drugs discoveries, only a handful of drugs have been discovered and be effective against malaria. So they have, but they are developing highly resistant against the Plasmodium. So current or the final antimalarial regime, artemisins, and with combination therapy, ACT approved in 2012. So artemisinin considered last druggable drugs against malaria. So malaria outbreak might be happen in the future, and so it can put billions of people at the risk because development of drug resistance against Plasmodium. So second part is a low anti-malarial drug diversity. As you can see, there's only 3 classes. All the drugs have been discovered so far. They only divided to 3 classes: acrylic amino alcohol, artifolates, artemisinins compounds. So Plasmodium developed resistance against all antimalarial classes. New antimalarial with novel mode of action are urgently needed. So third is the rationale of this study. Why we want to carry this kind of research to discover new drugs for antimalarial. So consider what we were using for our source of discovery is a microorganism. It's considered unlimited sources for bioactive compounds. Seconds, humanized GSK3 may be lead to identification of antimalarial agents. Third is a synergistic resistant development, an effective approach to identify antimalarial molecule targets. So as one is the first is microorganisms considered prolific sources for antibiotic compounds. 45% of biotech compounds are originated from microorganisms. Actinobacteria account for the 16% of the total compound. From the 200 genes antibacterial uses, Streptomyces contribute 60% of the bioactive compounds. As we know, the strain that we use H11809 also consider a Streptomyces species. So expression of other genus or consider a very active medicines or new class of compound is very urgent, maybe to a way to discover this kind of new drugs. Second is humanized GSK3 beta inhibitor. It's considered a potential target preliminary screening approach to identify antimalarial. GSK3 beta is a conserved kinase, can be found in its plasmodium, a validated antimalarial targets. Subtle structural differences, especially is ATP gatekeeper residue of GSK3 beta in different species, may affect compound specificity. So ATP getter of humans GSK3 beta is called leucines, but ATP gatekeeper for plasmodium GSK3 beta is maintenance. Affect the binding of Paulon's derivatives is then increase specificity toward a plasmodium GSK3. Selective of plasmodium GSK3 inhibition is possible. So what are the advantages targeting GSK3 for antimalarial development? Okay, this is a combination of human GSK3 and plasmodium GSK3. Human GSK3 involved in the dramatic increase of immune response upon the plasmodium falciparum infection, considered a cytokine storm. Cytokine storm responsible for fatal cerebral malaria. And essential for plasmodium GSK3, essential protein for plasmodium survival, involved especially during blood cell stage of infections, and valid target for antimalarial drug development. So this dual inhibition of host and parasite GSK3 acting as double-edged sword effects, prevent fatal plasmodium falciparum infections, and also is again plus inhibit plasmodium falciparum growth. So is expressed human kinase are routine used in University Malaysia Sabah to search for kinase inhibitor, includes humanized GSK3 beta. So FH028 also consider is a GSK3 beta inhibitor, a non-cytotoxic potential antimalarial, and H7607 and H7530 GSK3 beta inhibitor. I think it's non-cytotoxic, consider potential antimalarial, is consider anti-published data. So this data suggests that E-based assay expressing GSK3 beta can be attractive approach to narrow down sample for selection for antimalarial activities. So because this is easy to perform and comparative to in vitro plasmodium assay, and this is because this is very cost effective, and plus I think the attention is no ethical issue involved in using the research using yeast, unlike the using animal or humans. So third is a spontaneous drug resistance development to identify a unique mode of actions. So in nature, a plasmodium capable of delivering drug resistance via genetic mutation, as we know. This is how they develop a drug resistant against different type of drugs, and it could be mimicked in the laboratory. So lead to identification of new antimalarial drugs or drug targets, or plasmodium ATP4, plasmodium CARL, and this is a useful method to identify the molecular target or molecule of action of mode of action of antimalarial via a conventional in vitro assay. So this is the exposure of the weeks 1.5, week 3, week 4.5, and week 6 with exposure using the plasmodium 3D7 water cells. Okay, objective of this study first is to evaluate antimalarial activity. So the crude extract of with the humans GSK3 beta inhibitor activities. And to identify antimalarial compound produced by Malaysian soil microorganisms, especially the H10109 cell strain, Streptomyces strain, Third, to identify a mode of action of most potent antimalarial compound used in this study. So 5.0, this is a methodology used in whole research. So like you can see is the whole is a big project. So we use the bioactivity using preliminary screening using GSK3 yeast. Then after that we use reproducible tests in recants and optimize our mass production of strain and using extraction of heat stability tests and anti-malaria assay and using a malaria strain and others compound also. So after that we follow by purification identification of active compound using liquid extractions, and all the compound is using HPLC, others, then MMR. Also, then all the compound isolated by using a study of mode of action for study. So we're gonna use only the in vivo is base assay in silico study, ion dependency assay to determine the suitable ion, and these others some part is when we cover by the my talks today, because they will be using for other compounds. So for result and discussions, 6.1 yeast-based assay, as I mentioned just now, we use yeast-based assay as a potential preliminary assay. So assay to identify for anti-malaria drugs discovery. So as you know, yeast-based assay is very cost-effective, and it is possible no other issues. So we use H1180 streptomyces. Okay, you can show this here. GSK3 3 beta assay show in vivo inhibitions when testing on anti-malaria with plasmodium serum 3D7. Also, this is done in two places in UC Pakistan, Malaysia, and also repeated same test repeated in weekends. So it show a positive result. So a sample inhibit GSK2 beta also show anti-malaria activities. As you show, this is different type of strain or sample, and this is a strain that other isolated from. As you can see, there's 6 strain that show a positive GSK3 beta in vivo, also show against anti-malaria activity using a plasmodium 3D7 standardized strains. They have other 60 over strain that from the other species that didn't show the GSK3 beta result, so didn't show any result in the strain tested in recants. Overall, okay, sample with GSK3 beta individual exerted anti-malaria activity as well. Interestingly, non-assessed GSK3 beta activity sample have no anti-malaria activity, just as I show now with the 60 or 60 sample. Hence, yeast-based assay expressing GSK3 can be a good preliminary screening to narrow down sample selection for anti-malaria. So this is means it's more cost effective using than rather than you straight away do the cell culture assay, which is considered more expensive, and this is very limited. So this is very good. This type of assay, yeast-based assay, is very good to just start the screening when you have a crude extract isolated from microbe or other plants. I think this is a good preliminary assay just to do a screening. So the second type is discovery of active compound. Debuter phthalates, DBP, or we just DBP sources is isolated from as mentioned, H11809 from the Malaysia. So they show a plasmodium 3D7 IC50 of 14.4 micromolar. It's anti-malaria mode of action is really unknown so far. So it's have been reported a biological activity as antifungal, antimicrobial, and anti-malarious. So the other strain is nocardamines. It's also isolated from the H11808 and other strains. Plasmodium 3D7 IC50 show a 1.5 micromolar. Anti-malarious, possible ion chelating, but it's not confirmed. Reporting biological activities are considered anti-malaria. So a total of 18 compounds were identified or suggested from the soil of Malaysia and Japanese sources. So this is a mode of action, DPP, anti-malaria potential inhibitor, GSK3. Okay, this is using the exposure. As mentioned, using a different multiple exposure. Exposure 1, exposure 2, I follow exposure 3, and we increase the concentrations. So this is a 90 micromolar, considered higher concept possible, strength. Activity of against plasma 3D7 versus TBP 4. No significant drug resistance was developed. DB resistance development fails, therefore hence mode of action remain unknown, so it is still considered worth to further in other way of to identify of mode of action. So DBP molecular target is in this bill for but as we know for this study, although this mode of action remain unknown, but DBP molecular target is invisible for plasmodium survival, as you can show from the curve. So we space assay a complement also in silico study. So DBP selected inhibit humanized GSK3 beta in vivo. So as an this is a use sample yeast sample, so inhibitions on different temperature, 28 degrees and 37. So DBP is using 160 microgram per disease. It show no inhibition on the 28 degrees, but it show 10. This is considered millimole inhibition. So this is called a selective GSK3 beta inhibitor. But nocanamines as other candidate also, but show no inhibition in the 28 and 37 degrees. So TDZD also show a selective GSK3 inhibitor, and H766, one of the strain also from the Malaysian soil, isolated from Malaysia, also show a non-selective when it show an inhibition on the 28 degrees and 37 degrees. So non-selective GSK3 inhibitors. So DBP binds to the humanized GSK3 beta active site, forming a hydrogen bond, the glucosamine 97. So glucosamine 97 is considered a target selectivity, increasing because 97 is a conserved amino acid subunit for the plasmodium GSK3 as a glutamine 20s. So plasmodium GSK3 knockout PF strain was reported to be unsuccessful, collaborated with the ancestral DBP resistance strain generations. So DBP might be targeting plasmodium GSK3 as its must be anti-malarial mode of actions. So second part is nocanamines. Nocanamines is known ion chelators. So the additional of more than 12.5 micromolar ion reduce more than 90% of nocanamine efficacy. So this experiment results show that confirm the combination as a staff plasmodium from these ion sources. So overall, the findings: DBP possible inhibit of plasmodium GSK3. Nocanamine confirmed as now it's confirmed as a ion chelating, starving plasmodium from its ion sources. So for the conclusion, sample with humanized GSK3 beta inhibitor activity also exert anti-malarial activity. Hence, yeast-based assay can be interesting preliminary approach to do a discovery. So this is very useful when you are start up a low cost when what for drug discovery. Second, a total of 18 compounds were identified, suggested or from Malaysia or Japanese sources mechanism, include compound never reported as anti-malaria or possible new drugs compounds. Microbes should be further explored for anti-malaria drug discovery. So this is a reference may be used for my presentation and the whole studies. So I want to acknowledge this research is mainly supported by FRGS, Fundamental Research Grant from Malaysia's governments. And Mr. Fauzi, the PhD student, is supported by scholarship from the SLAP scholarship from University of Malaysia Sabah, and also the research attachment from the International Program Associate IPA from Weekends. Thank you very much for your attention. Thank you.